Very good evening to all of you. I welcome you all for today's webinar on what do the experts say. And today we have an interesting topic, prevalent treatable intellectual disabilities, how genomics can make a difference. This will be the last talk in our series on genomics in pediatric neurology. From next week onwards, we'll be having the SIGs taking over, and for the next 4 Wednesdays, we'll be having activities from the neuromuscular SIG. Today's talk would be delivered by Dr. Binu Kumar. Dr. Binu Kumar is a principal scientist in the Division of Genomics and Molecular Medicine at CSIR Institute of Genomics and Integrative Biology, New Delhi. He is Associate Professor, Faculty of Science in ACSIR, India. Dr. Binu Kumar has co-authored more than 50 research articles in peer-reviewed journals, and his main focus of his current research is on development of cell and gene therapy for hematological diseases. He has got many honors in his name. DST ACRB Early Career Research Award in 2017, Exceptional Summer Internship Program Mentor Award in 2016 by the NINBS USA, and the recipient of Professor R. Nath Gold Medal Award from PGIMR Chandigarh for the best published research work in biomedical sciences during the year 2010 to 2011. To discuss this topic, we have got 3 experts. Dr. Jitesh Puten is Associate Professor from Hamad bin Khalifa University, Qatar, and he has done a PhD in bioinformatics. He also got many awards. To name a few, he is a member of Congregation University of Oxford, UK, Best Poster Award at Irish Association for Cancer Research in Galway in the year 2010. He has got PhD scholarship from Queen's University, Belfast, UK, and the Best Poster Award in the year 2001. Next. We have Dr. Sanjay Pandey, who is the Professor and Head Department of Neurology and Stroke Medicine at Amrita Hospital of Faridabad, Delhi NCR. Sir has got more than 200 publications, and his key area of interest include botulinum toxin, dystonia, tremor, functional electrical disorders, and post-stroke movement disorders. Sir also has many awards, to name a few. Then we have Dr. Mary Ibe. Sir has got the Indo-USA Science and Technology Forum Fellowship, and he has a fellowship of IAM. Next. Dr. Mary Ibe is a professor and head Department of Pediatric Neurology from Government Medical College, Trivandrum. Ma'am is a regional director and visiting professor at the Institute for Communicative and Cognitive Neurosciences. Ma'am has done her MD and DM, and she has MNFs and FRCPCH degrees also. She has published several articles in national and international journals. She has had the Best Research Award from IMA AMS Kerala in the year 2018. Ma'am is a first-time holder and gold medalist during MBBS, and she has been the past president of Kerala Neurochapter of IAP in the year 2012 to 2014, and treasurer of IAP Neurology Kerala chapter, and she is executive committee member of NSI Kerala for the year 2014 to 2015. These 3 experts will be discussing this topic. I would request Dr. Binu Kumar to take over.
Is my slide visible?
Yes, it's visible, Dr. Binu.
Yes.
So welcome to everyone. Thank to the organizers, especially Dr. Shifali Madam and Dr. Magesh, giving me an opportunity to present some of the work we do here at IGAB. So today the title of the topic is Prevalent Treatable Intellectual Disabilities, How Genomics Can Make a Difference. So before I slides, okay. So my lab here at IGAB, I basically works on genomics and biology of neurological diseases. So I will be touching up some portion of the work we do here at IGAB during my talk. So I know in this audience, there is no general introduction needed for intellectual disability. So what I like to do is directly go to the genomic aspects of intellectual disability. So, when you see the spectrum of genomic aspects of intellectual, so 50% of the genomic aspects is not known, and 15% of this ID comes under the chromosomal abnormalities, and again 15% comes under copy number variations, 10% is for point mutations, and again the last 10% is for the mutation in the telomeric rearrangement. So it can be some of the IDs are monogenic, so it comes under the category of dominant ID or recessive ID or X-linked ID. And major portion or chunk of the major portion comes under the inborn neurological metabolism. And so one of the major problem for the diagnosis of intellectual disabilities, it shows overlapping symptoms, also it shows the comorbidity. That was a huge challenge in the diagnosis of the ID. Gee, I have a problem for moving the slide. Okay. So, so when you see the spectrum of mutations, so there are different databases are available for the different IDs. So when you see that Malachite's database, there are 500 clinical variations are associated with the ID. So that implicates almost around 2,715 genes are implicated for the ID. There is another database that is called OMIM. In that database, they reported more than 750 autosomal genes are responsible for the IDs. I am facing a problem to move my slides.
So when you have shared your screen, on the bottom left of your screen, there'll be arrows which you can use to move ahead. We have moved one slide ahead now.
It's not moving from my side. Okay, so so when you see the diagnostic era, so ID can be, you can divide it into two different parts: pre-genomic era and the post-genomic era. So in the pre-genomic era, the diagnostic yield is below 20%. But in the case of post-genomic era, there are multiple techniques are available. Say, for example, G-banding, chromosomal microarray, whole exome sequencing, whole genome sequencing. So adopting these techniques, the diagnostic yield can be increased to 43 to 47%. When you see the nature of the mutation, majority of the mutations are large-scale chromosomal aberrations, copy number variation, and single nucleotide variations. So there are multiple studies, multiple cohort studies available across the globe, and their diagnostic yield vary from 21% to 42%. And again, in India, there are multiple different individual studies are available. Say, for example, in the case of Jain et al., published a paper in 2013. So according to that studies, 62.1% of their major cohort, the genomic aspects is covering the major cohort. And in the case of Johri et al., so prenatal and perinatal and neonatal cases are predominant in the case of IDs. And in case of Sao et al., 2014, so perinatal cases are belong to the predominant dominant factor. So there are multiple studies available. So it can be, so the prevalence is vary from 1% to 3.2% in the population, depending upon how they are defining the prevalence, how the study population, and how the study design and how they measure to identify the ID. So if you take a whole, so the variation from, it can happen from 0.2% to 20%. So that is a great thing, but the problem is, so that huge variation that doesn't help to planning a precious policy, or if you want to deliver a program, that's for the neonatal program. So that studies are not helpful. So my take-home message is, till now, there is no large-scale cohort studies are available in India so far. So we need to have a large-scale studies are wanted. So what are the challenges actually? How we can so what are the how we can diagnose the IDs? So chromosomal microarray, till now recently people are referring for the first tier of diagnosis. But according to the Lansstein 2019, so he proposed the whole genome sequencing as the first line test for comprehensive capture of all the diverse spectrum of disease causing variations in intellectual disability. So what are the advantage of whole genome sequencing? In the whole genome sequencing, that overall diagnostic rate increase in the double compared to the clinical microarray. And it can capture structural variation where it can capture the structural variation. Not only is that, if you have whole genome data, the same data can be used for the analyzing the single nucleotide variations, UPD or STR detections. So according to Luther et al., whole genome sequence can provide overall diagnostic yield 47.9%, 47.6%. So how we can prioritize the variants? So there are multiple ways we can, if you do the whole genome sequencing or a whole exome sequencing, there are multiple ways we can prioritize the variants. So one method is we can look for the deleterious exonic variation. From the deleterious exonic variation, we look for the rare variant, so that we can use for the population genomics data. And from the rare variant, we can look for the literature curations, the gene filter. And from the gene filter, we can also use different computational pipelines like SIFT, polyfin, CAR, and we can identify single gene. And if you identify single gene, we do the ACMG AMP guidelines. But the problem is still this whole genome sequencing is a expensive affair. It will also take a time consuming. But in the case of microarray, so it's a signal intensity file. We can call for the copy number variation, and clinically relevant copy number variation can be captured, and we can do the ACMG AMG guidelines. So one of the problems with the chromosomal microarray analysis is CNV interpretation is challenging. In the case of mutation spectrum, there is no comprehensive clinically relevant variants database are available so far. So when you see a subset of ID, it can be considered as a treatable IDs. So in treatable IDs, there are 1,100 disorder caused due to the mutation 137 genes. So most of them belongs to the inborn error metabolisms, and some of them are single gene disorder, but that is not screened for frequently. And some of them are classified as a prevalent IDs. So one of the best example is, so recently 2019, the Singapore genome sequence is completed. So in there, what they found that there is an unexpected higher frequency, that is 6%, for the 13 of the inheriting disorder. So that is a extremely higher frequencies. So that if you get that kind of data, that data can be used for the newborn screening, or we can plan to we can prioritize the treatment programs. So what are the clinical challenges in case of IDs? So the clinical diagnosis, phenotypic heterogeneity. So, and is misdiagnosis to many other diagnoses. And genetic diagnostic difficulties, genotypic heterogeneity, no single database available for all causal variation implicated in IDs. And other thing is in concern to the India, so population specific variation. So limited Indian population specific studies or knowledge about prevalent or founder or hotspot variation is available. And treatment driven diagnosis approaches are very limited. So based upon this prior knowledge, one of our objective was to understand the genomic basis of idiopathic ID or GDD using an independent patient cohort across India. So what I'm going to explain is there are two different studies. So there are multiple hospitals are involved in this study. So the Dr. Shefali Gulati is one of the major contributors in this cohort. So AIIMS, then a hospital from Jaipur, hospital from Jodhpur, and Tirunelveli Medical College are the main contributors in this study. The number of probants recruited for is 67. Total number collected for this and from this entire study is 200. So the provisional diagnosis, it's a varying diagnosis like ID, GDD, MAR, hyperactivity syndrome, and intellectual disability. And there are different kind of clinical presentation that starts from subnormal intelligence or autism spectrum disorder. So based upon this, when you see the patient distribution or proband distribution, so 37% of this probands belongs to, male participation, and 62.5% is male, and the 37.5% is the female participation. So the left side you can see the distribution of provisional diagnosis, different cases. So when we initially the differential clinical diagnosis, and we all of the patients recruited are put for the whole exome sequencing. then this whole exome sequencing mapped it to the referential genome, then call for the variants and then prioritize the variant. And once we prioritize the variant, segregation studies are done in the family using Sanger sequences. So I don't want to explain the pipelines. What are the pipelines? Are normally what is the pipe? Once you have a VCF file, from the VCF file we can use ANOVA to remove the synonymous variations. And again, you can use the population frequency filter and computational 2, and then use ID gene filter based upon the literature. There are different ID genes previously annotated associated with the individual ID. So around 825 genes are associated with the individual ID. Then again, we can use a subset of filter, that is treatable IDs, that 125 genes, based upon the literature survey, there are 125 genes are associated with the treatable IDs. So I will summarize the A series, the cohorts. Total 67 probands are recruited. So in 67 whole exome sequencing is done. So in 30 probands which have a putative variants are found. In this 30 probands, 6 probands reported with the pathogenic and likely pathogenic non-variations, 20 probands reported with uncertain significance, 4 probands reported with the conflicting difference, and 10 probands with varying in the TD genes. So the remaining 37 probands, so yet to be unsolved. So out of 32 out of 37, 32 probands which there is no variation we could prioritize, but 5 probands which could multiple genes and multiple variants are needs to be prioritized. So when you see the, I will take on one example from this cohort. So this is the case referred from Dr. Shantaram, Tirunelveli Medical College. It's a male patient, a 7.5-year-old. There is no consanguineous marriage parentage, so no birth asphyxia. So the delayed in motor and mental development, and so the clinical diagnosis probably inborn error metabolism. When we do the whole exome sequencing, we could able to identify a mutation in ATP7A gene. So when you see the ATP7A gene, that is previously associated, it is not associated with Menkes disease. So this is a non-pathogenic mutations, and we could able to solve this case. And second one is it's a probable type of leukodystrophy. This is also the case referred from Dr. Shantaram from the Tirunelveli Medical College. This is a 15-year male, again non-consanguineous parentage. So full-time normal delivery, delay in both motor and mental disorder, and delay in social and language problems. So when we did the whole exome sequencing, we could able to get a mutation in the gene ABCC8. So this gene is associated with the hyperinsulinemic hyperglycemia. So it's a heterozygote. Inheritance is autosomal dominant. It's a non-pathogenic mutation. So the summary of this case series is what we could able to find now. This is the whole exome sequencing could unreveal mutative genetic variation, 40% of the intellectual disability patients. 55% of the cases still remaining unsolved by using whole exome sequencing approach. This have been prioritized for looking for the chromosome number. Looking at CNV from whole genome. Array-based whole genome analysis, or we can also look for structural variation in the whole exome sequencing. So treatable conditions or ending diagnosis or DASI to management of options account for 33% of all identified mutation, all identified putative variations respectively. So the second is, we would like to estimate and understanding the carrier frequency of ID disorder focusing on treatable ID using a population scale genomic data sets of global population. So we all know that there are multiple population genomic data available. It's starting from the release of the whole genome project. So there are multiple genome projects are conducted from 2001 onwards. So here I am going to summarize 3 population genome data. One is from our own Indigene data, and second one is for the Singapore, and third one is the Malaysia genome. So in Indigene, so previously we use the foreign data for the baseline data. So we don't have any baseline data for the Indian population. So what we planned is that we sequences around 1,000 individual. This is apparently self-declared healthy individual. So it comprises 50 samples per state. The 26 states are covered. So we did the whole genome sequencing. And the outcome of this data is then from the whole genome sequence, we look for the treatable IDs. So there are 125 genes associated with 116 treatable intellectual disability disorder. So 100 were autosomal recessive conditions, 8 were X-linked condition, 6 were autosomal dominant condition, 1 was associated with the mitochondrial disorder, and 2 were described both autosomal recessive and dominant in nature. So the pipelines are, there is a 1029 apparently healthy individual, whole genome. Based upon the literature mining, we prioritize the 125 treatable IDs. From 125 treatable IDs, we look for the ClinVar and with pathogenic, likely pathogenic variations. So then computational pipelines we use for different computational pipelines used for the prioritization of the variant. So the take-home message from this is 24 variations are pathogenic mutations, 44 are likely pathogenic, and 11 are benign, and 230 belongs to the variants of unknown significance. So why we need to do the ACMG criteria? So ACMG criteria is a standard tool. It comprises 28 attributes. So we can grade the variant when a variant is passed through these 28 attributes. So this some of the attributes involves population database, then functional studies, segregation studies, and computational tools, and to a larger extent some case control studies. If you pass through a variant to this different set of attributes, at the end we can classify a particular variant into pathogenic, likely pathogenic, or variants of unknown significance. That is the advantage of the ACMG AMG parameter. So when you see this, our indigene populations, so there are almost 3 lakhs variations are associated with the treatable ID. From these 3 lakhs variations, we look for the ClinVar variations. There are 30,000 are ClinVar variation associated. From the ClinVar, so there are 211 variations are associated with the pathogenic and likely pathogenic. And on the other side, we can actually filter out non-coding intergenic variations, and again filtered out major allele frequency less than 1 allele frequency. And we also filtered out the benign variations using computational tool. So based upon this, we could able to get a 2,900 variations associated with the treatable ID. So when we did the ACMG AMG criteria, so the take-home message from this indigene data is 68 pathogenic and likely pathogenic variants are associated with this. We could able to get it from the 1029 Indians. 36 genes are 36 genes. Then 34, most of them are in 36 genes and diseases, and 34 are autosomal recessive conditions. 93 individuals are heterozygous individuals. These are carrier for these conditions, and 1 in 11 individuals in India carrying pathogenic or likely pathogenic for 36 treatable IDs. And these are the sum of the genes associated with the treatable IDs. So what we able to get is that 13 variants common in 2.5 times more in indigene than South Asian genome. So some of the variants belongs to the variants that are associated with the Wilson disease, PKU, or BTD deficiency. I will explain Wilson disease later in the coming slide. So this is the representation. Now we can representation of our indigene population compared to the 1,000 genome or other Genome Asia or other population database. So then from the prevalence data, we can also, from the frequency, we can also find out the prevalence of some of the variation. So these are the sum of the prevalent gene, and you can also find out here you can see the prevalence of these diseases from the Indian population. So the summary of this indigene project is, since the newborn screening is not performed for most of this disorder in India, the majority of the case that could be prevented at the early stage are left undiagnosed. Looking at the high carrier frequency from 1029 apparently healthy individual data, it is very clear that the prevalence of this condition is quite high. And given the consanguinity status of India, also keeping in mind that most of these disorders have autosomal recessive form of inheritance, it could be probably leads to very high number in case of it suffering from ID. However, if this disorder can be potential implication in newborn screening, can be detected at an early stage, significant therapeutic intervention can be made as possible. So coming to the Singaporean sequencing Malay projects, it's a 100 individuals. The pipeline is similar. So the take from this 100 individuals, I mean Singaporean sequencing Malay project. So what they could able to find is that what we could able to get from this treatable IDs are 2 pathogenic variations, 17 likely pathogenic, 1 benign, and 2 likely benign, and 78 belongs to the VUS variants of unknown significance. So this is the pipeline we use. When you look into the data, so there are almost 35,000 variations are associated with this treatable ID. From this variation, when you look for the ClinVar, so almost 14,000 variations are associated in the ClinVar database is associated. So from this, when you see the pathogenic and likely pathogen, 52 variations. And from the 52 variation, and we also filtered out most of the common variation and most of the non-pathogenic variations or benign variations, and we got 100 variations. From this 100 variation, we did the ACMG AMG guidelines. From this 100, so what we were able to get is 19 pathogenic variations are associated with 19 pathogenic variations are presenting in the 100 individuals, and 15 genes and diseases are associated with this one. 24 heterozygous individuals, that means 24 are 20. This is the carrier frequency, and 1 in 14 individuals are individuals in SSMB carry pathogenic or likely pathogenic for 15 treatable IDs. So in the case of Singapore sequencing Indian, so basically this represents the most of the South Indian population that they are living in this part of the country. So the pipeline is again same. So when we did the variant prioritization, so almost 26,000 variants are associated with this intellectual disability. And from the ClinVar, again, 12,000 variations. And when we narrowed down to 54 variations, from the 54, so 3 pathogenic variations and pathogenic or likely pathogenic, 3 genes and diseases are associated with this condition, and 5 heterozygous individuals, and 1 in 7 individuals are associated with this, some of the common treatable IDs. So the summary is, we aim to build a diverse map of treatable ID variant in Southeast Asians, Malay population, and Singapore population using deep whole genome sequencing of healthy South Asian Malay individual and 38 Singaporean individual. So we investigate the allele frequency of pathogenic and likely pathogenic analysis revealed 19 and 13 pathogenic and likely variations in Malay and Singaporean population, respectively, with translated into 1 in 4 individuals that carry 15 treatable IDs in Malay, and 1 in 7 individuals of carry for 3 treatable IDs in Singapore. So when we did the whole gene, I mean in Indian genome gene, so one of the most prevalent disease what could able to get is the Wilson disease. So in Wilson disease again is an autosomal recessive condition. It's due to ATP7B gene. So it has a predominantly three clinical presentation. One is neurological complication, hepatic complications, and psychiatric complications. So there are multiple studies are available for the Wilson disease across the globe. Since the carrier frequency rate is 1 in 90, and the carrier rate is 1 in 90, and the global prevalence rate is 1 in 10,000 to 1 in 13,000. But unfortunately, in India, we don't have any pan-India scale, Indian allele frequency data available so far. So when you see the nature of this is a monogenic disorder. So the gene associated with this Wilson disease, ATP7B, it's a copper exporter. So when you see the nature of this protein, it has a copper binding domain, it's a transmembrane domain, ATP binding domain. So there are multiple mutations are reported across India, but there is no pan-India study and there is no systemic annotation done for the Wilson disease. When I started my work, so what we initially did is we collected all the variations reported so far. So we could able to find 4,755 variation associated with ATP7B. So from this 4,700, when we did the filter checking, so there are 1,799 are unique variations, and this 1,700 unique variations we did for the ACMG criteria. From you can see that 54% of the variants are belong to the VUS category, and 25% are belong to the pathogenic category, and remaining 7% belongs to the benign. So when we look for the nature of the variations, so most of the variations are concentrating on certain exons. So and these are the sum of the exons. We can see predominantly these variations are pathogenic variations. So this is the when you look into the variations are reported in India, it's also representing some of the exon shows most predominant variations. So we can map this variation into the proteins. What we found that the pathogenic variations are presenting in the functional domain of the protein. So that cause the functional functionality of the protein. So when we look into the global scenario of this Wilson disease, the most number of cases are reported in China, and India is holding the second position. So this is the allele frequency for the ATP7B gene in different population. Here you can see that some of the variants are predominantly presenting in certain population. The advantage of this study is we can use this certain alleles, certain variation for the large scale screening. So in India, we also in Indigene in 1029 individuals, we also look for what is the nature of this, what is the allele frequency and carrier frequency. What we found that the carrier rate, according to the Indigene data, the carrier rate is 1 in 69 and the prevalence is 1 in almost 20,000. So based upon this, we build up a database. So the advantage of this database is we reported all the variations, and if a clinician now find a variation, they want to know that whether the variation belongs to pathogenic or likely pathogenic. They don't want to do any computational analysis. They don't want to do any ACMG. They can simply type the variant into the database. Then that database will tell you whether the variation belongs to pathogenic, likely pathogenic, or variants of unknown significance. So these are some of the examples of the database. So the next portion is, as I explained previously, there are individual center Wilson disease studies are available, but there is no pan-India study available so far. So we actually created a consortium called Indian Collaborative Research Consortium on Wilson disease. Around 22 clinical hospitals across India is part of the study. So we could able to recruit almost 125 patients across India. So the summary of this, again, the patients we recruited. So we exclusion-inclusion criteria is based upon the modified Leipzig score. So some of the patients were not included in the study because that does not qualify this modified inclusion criteria. So we did the whole exome sequencing for this Wilson disease, clinically identified Wilson disease patients. So and then we variant prioritization. So I will explain one of the representative case, then I will summarize the study. So this is the patients recruited from Dr. Sanjay Pandey, GB Pant Hospital. So the patient is showing dystonia and high level of SCPT and SGOT level, and it's a left-handed dystonia. Mouth opening and left-hand dystonia, and KF-ring positive. So we were expecting a homozygous pathogenic mutation, but we could not get any homozygous pathogenic mutation, but we could able to get a mutation, heterozygous mutation, the first mutation in exon 2, and the second mutation in the exon 18. So then this whole exome data we validated in the Sanger, and we can also see that the one variant is coming from the father and the other variant is coming from the mother. So it is a classical case of compound heterozygous. And the sibling is a carrier for this condition. So this is the summary for the entire spectrum. So what could able to get is the 15 ATP7B positive homozygous mutations, and 24 ATP7B heterozygous mutation, and 11 ATP7B negative compound heterozygous, and 11 ATP7B one heterozygous variation, and we could able to get a novel 22 mutations from the entire study. So what could able to identify is that some of the variations are, let's say for example, C27X is a highly prevalent compared to the other variations in our population. So one of the other thing is there is a 40% of our clinically identified Wilson's, but when we did the whole genome sequencing, we could not get any variation in the ATP7B gene. So we are interested to see if there is any other genes responsible or any other gene associated with the Wilson disease. So we did a burden testing using indigene as a control population. So in quintile analysis, what we could able to find is there is a 4 other genes can also came up with the analysis, but this is the preliminary analysis. We needed to do functional study to showing that these 4 genes also responsible for the Wilson disease. So based upon indigene data and ICRO and Wilson gene, now we could be able to identify hotspot mutations. So this hotspot mutation can be used for the initial screening. Okay, so we also looked for the structural variation in our cohort. We could be able to get a one structural, there is a 332 deletion, and this this variation we actually confirmed with the Sanger sequence. And during COVID times, so what we develop, actually we develop a scalable cost-effective approach. The approach is like, say, when you see the entire ATP7B gene, there are 28 amplicons. These 28 amplicons can be put in a single tube, so we can sequence it in a single reaction. So we standardize this protocol, and you can see the coverage of the each and every amplicons, exons, and this high throughput is validated in patient cohort and validated in patient cohorts. So it can capture the amplicon faithfully, and we validated in this techniques. We can also validate in the Sanger sequencing method. So one of the other problem in our cohort, not only in the cohort, in most of the clinician facing is when a clinician refer a case to genome study, sometimes it's tend to be the variants of unknown significance. So if a clinician get a report saying that variants of unknown significance, it's a dead end. So can we do something related to the variants of unknown significance to the variant? So what we use is, in case of Wilson disease, I will explain with some of the examples. So we use an alpha fold because ATP7B protein can be modeled, and this can be, and the variation also can be modeled. So initially what we did is wild type and the non-pathogenic mutation, we model it and see whether in using the alpha fold, whether this mutation actually representing, once you do the stimulation, whether that mutation is showing pathogenic, and deterioration in the protein structure. So what we able to see that some of the non-pathogenic mutations compared to the wild type using alpha fold, it shows a structural deterioration. So next is this mutation, we can do this, we can clone and we can do the site-directed mutagenesis. And once you make the site-directed mutagenesis, you can overexpress this mutation in a cell line. But the cell line needed to be have ATP7B negative. So what we did is we take a HEK cell and deleted the endogenous copy of ATP7B using CRISPR-Cas9. So we create a knockout ATP cell line, HEK cell line, then overexpress this mutant, wild type and the mutant, and then did the copper export study. So what you can see that compared to wild type, the previously non-pathogenic mutations shows decreased copper export activity. So that we can say that our methodology is working and standardized. So now we can use the same methodology for the VUS variant. So what we did is, again, we modeled the VUS variant. And here you can see some of the examples of the stimulation studies. So it may be mild deviation or a high deviation. So depending upon this stimulation study, and we can also see this residual fluctuation in this model. And this is the summary of SIFT, polypin, and CAD. Then what we did is those VUS cloned into a vector and did the SDM, and this side directed mutagenes and this VUS variant overexpressed into ATP7B knockout cell line. Then you can do the copper export assay. What we found that this VUS compared to the wild type, it shows deficiency in the copper export. Not only that, the cytotoxicity of these mutants are very high. In addition to that, it also shows a lipid accumulation. And this mutant also shows enhanced apoptosis compared to the wild type in presence of copper chloride treatment. And you can also see the morphology of this mutant also changes. And you can do multiple other parameters, like we can measure the half-life of the protein. So some of the mutant shows less half-life compared to the wild type. So, and the other way how we can validate this protein is, when you see the nature of the variant, so normally if a ATP7B is present in the ER, in the presence of copper, it moves to the ER to the lysosome and then to the excrete the copper lysosome and finally to the vesicles. So what happened at the time of mutant? So most of the variant, I mean pathogenic mutation or likely pathogenic. Let's say in the case of this is the one of the VUS variant. This variant present still retaining in the ER. It is not moving to the lysosome or not moving to the vesicle. So that means the trafficking is affected for this particular variant. So like that, we can actually functionally study a particular VUS, whether it affected the functionality of the protein. So it affects some of the variants, it's still stay in the endoplasmic reticulum. It's not moving to the ER, and it's not even moving to the lysosome. So the summary of this functional study is, here you can see that previously some of the variants were classified as a benign, but when you do the functional studies, these variants or variants of unknown significance not actually a benign or not VUS, it's reclassified into a pathogenic variant. So that is the beauty of if you do the functional study. So we can reclassify the VUS based upon the functional data. It can be comes under pathogenic or likely pathogenic. And some of the variants still after the functional study, they still remain in the same as VUS. So to the last stage, one of the problem with the Wilson disease is some of the patients undergoing the depenicillamine triangle, they actually adverse drug reactions. So what we can do with whether we can define a precision medicine. So the idea is, if we take, because the mutation is known, okay, if we take a patient blood sample and when you can differentiate that blood PBMC into an iPSC, and from the iPSC we can again reprogram into, we can characterize the iPSC and we can correct the mutation in iPSC, and this corrected mutation we can do the functional validation, and later stage it's our dream. So we can actually once you corrected the iPSC, so the idea is to inject it back to the patients. So what we did is, so this is the patient's shows adverse drug reaction in the depenicillamine treatment. We took up the cases. So we generated an iPSC, induced pluripotent stem cells from this patient, and from the iPSC, we characterized the iPSC. So this iPSC shows pluripotency. So you can look for the different marker, pluripotency marker. And then you can actually see whether that iPSC shows again trilineage differentiation. So it will be able to differentiate it into ectoderm, mesoderm, and endoderm. So the idea is, now we have a patient's sample and patient specific iPSC. So the idea is, now we also clone some of the genes, some of the guide RNA, and try to correct the mutation. And after correction, so we would like to de-differentiate this iPSC into hepatocytes, and our long-term goal is to, so once you can able to generate a clinical grade iPSC, we can inject it back to the patient. That is a very, like, I mean, there are multiple stages that need to be done before going to the clinical trial. So that is our long-term plan. So what we have, Wilson, till now we have is, so we have one of the largest ACMG AMG classified for ATP7B gene. So we also have a cohort Wilson disease cohort, around 125 patients or whole exome, 125 patients suffering from Wilson disease, and most of the we did the whole genome sequencing for this 125 cases, and we could able to capture the spectrum of mutation from different parts of India. And we could also able to develop a high throughput, low cost diagnostic panel. In addition to that, there is a functional assays are available for validation of the, not only VUS, there are other functional assays available for the validation of other VUS gene as well. So, and last, and we also trying to develop a precision medicine for patient-specific iPSC for the future precision cell therapy. So, in addition to that, what we have is we would like to develop. We are developing actually different pipeline. So, it's saying that from genomic data to annotation to output, clinical output. So, there are different clinical annotations tools are available from VCF to then there is an automated tools available at IGB website and at IGB also. So we can actually use this clinical tool for the characterization of the particular variant. So I would like to thank, this is my summary slide. So most of the work it's from the Arishri, Vinod and Sridhar PhD student, and the Mukesh is my PhD student. So, and I would also like to thank to the Sridhar and Vinod for beautiful collaboration. And I also thank Guardian and the iCrowd family, and plus all the patients and families. And funding received from the CSIR. So thank you. It's open to.
Thanks, Dr. Binu, for your wonderful presentation. I'd like to take comments from our experts now. Dr. Sanjay sir.
I think Dr. Binu Kumar presented a very nice talk, and I have worked on a couple of projects. One is Wilson's disease, and the data he has shown is very, very important, at least in the Indian context. The reason is that many times Wilson patients who present with neurological manifestations, they are challenging to diagnose. And I think with Indian data on genetics, we are going to have our own data, which is lacking currently. So we have collaborated with in some other areas also, like in dystonia and dystonia Parkinsonism. And we are planning to collaborate with Dr. Binu Kumar's lab in patients with neuroacanthocytosis also. So this data is going to have a good insight regarding the genotype-phenotype correlation, what we are looking at Indian cohort, which is lacking. So this is a pediatric meeting, but as an adult neurologist, I can see that many patients of pediatric age group, when they grown up and they come to the clinic, they have a diagnosis of CP. But when you see them clinically, they have some phenotype like dystonia, or maybe some phenotype like Parkinsonism, and then you realize that that is not CP. That is some gene associated phenotype. So this data is very important, and I think we should collaborate more and provide more data for the Indian community. Thank you, Binu.
Thank you, sir.
Dr. Jitesh Muthen.
First of all, thank you very much for inviting to be on this panel. So yes, Dr. Sanjay said it's a very interesting presentation, and a broad spectrum of studies actually mentioned in his presentation. So it's like a lot of work that I can see coming from the case control kind of studies, looking at specific cases and the patients and their families. So doing the sequencing and trying to identify the genetic variants associated or causing the specific intellectual disability conditions, to the population kind of spectrum, end of the other spectrum, which is the population study, from the monogenic studies to the population studies. So from population genomics side, I would say actually, yes, again, there is quite a lot of resources available which we can tap into and identify these variants in these genes which are already known to be causal in intellectual disability, and how we can find novel pathogenic or likely pathogenic variants in those genes, and which will then help us in estimating the carrier frequency in the population. So it's a great effort that is being done by the IGAB and also the clinical collaborators across India who are looking at identifying the cases and also recruiting the patients, as well as this, as I said, the population genomics study where there is quite a lot of apparently healthy individuals whose data or genome has been collected, and which is also helping in defining the carrier frequencies in the population and potentially identifying novel pathogenic variants. And also, Dr. Binod presented the other end of these studies, which is the functional characterization of the variants, which is again very important. Of course, when we try to look at large number of patients, then we end up identifying a lot of variants of uncertain significance, which need to be explored further. And that's what is done in the last set of slides that Dr. Binod presented using various functional genomics analysis. And as I said, this is like a broad kind of presentation of the study on the study of intellectual disability. Thank you.
Dr. Miriam.
That is a very interesting talk. You have done some functional studies in patients where you found a VUS. Do you think other people can reproduce this, do this in other labs too, or can it be done only in an Apex lab?
Yeah, it's a lot of resource also needed. It's again depending upon which functional study we are looking for. In certain cases, let's say the transporters are there, so then we can easily plan functional study. Or if it is a mutation affect the structural changes. But certain, let's say, a lot of dystonia genes, we cannot even do the functional study because the readout is the major problem. So if the readout is there, so then we can plan the functional study. But some of the VUS we cannot do actually because we could not have any possible readout. So it's again depending upon which variant you are looking into, and if the readout is there, we can do the functional study.
From your studies on Wilson's disease, what's the population prevalence?
So it's
of carrier?
So that 1 in 69.
1 in 16.
1 in 69. Approximately 1 in 70. Yeah.
70. Okay. And have you done any studies on Menkes kinky hair disease?
Yeah, we also have a database for Menkes. I didn't present that database. But population-based studies we don't have. But there are 600 variations associated with the Menkes. So we did the ACMG, AMG guidelines also. That database also freely available in our IGB website. So there are different case studies are there, but there is no pan-India study available.
Okay.
Dr. Binod, Dr. Binod, will you like to speak something? And anybody wants to ask any question can raise their hand or write in the chat. Or anybody wants to share anything, their experience or anything, can raise your hand, please. Dr. Binod, do you want to speak something, please? Dr. Binod? I think it's important right now to understand, and the genomics are something which are the current and the future. The only thing is that, as Dr. Binod has said, ultimately it's going to come into an whole genome thing. And just wanted to share that it's very important to, very important to understand. Doing is, not that important, that difficult, I think. It is difficult, but obviously more difficult is to have the bioinformatics part and trying to understand and make sense of the thing. That's why earlier in the talk when Dr. Vinod was talking on the personal genomics, whether the personal genomics is going to be there of the future, which is going to come up after a couple of years. So we decided that we'll be launching a platform. So anyway, we have an AIMS IGIB, AIMS Center of Excellence collaboration with the IGIB for the last 5.5 years, in which we have been coordinating and collaborating with Dr. Vinod predominantly and his colleagues. We have planned to launch a platform, and that is being made, and it will be in the next couple of weeks we'll be launching it, and then it can be used widely, in which any clinician can upload any genetic report which is a VUS or anything you want to discuss about it, and then it will be discussed with the group of genetics people from the bioinformatics side. So we will be having a core group of the genetic side as well as a core group of some pediatric neurologists, and we would be looking forward for volunteers who would be wanting on either side, and then we can be discussing and then sharing so that we make sense of all what it is. It's not only doing is going to be simpler, because now you have so many labs coming up, mushrooming all over. And I also feel that maybe Dr. Vinod, we may like to have a QC of the the lab thing before we have that thing uploaded. So maybe we'll develop something in which there is some quality control, because there are some small labs coming up in that, to understand whether the quality is okay. And then other thing will be the bioinformatics part that we will be launching soon. I think it is important to understand that this is the way forward, and so many things were so many conditions were mimicking so many things. Now we are understanding that, in CP, there were so many mimics, and there's so many other precision medicine which is coming up. And ultimately, I think we will go towards genomics, going for pre-implantation, genetic screening and diagnosis in many of those familial cases. Also through IVF, I think that is the way it's going to go. But obviously, clinical, one has to remember that clinical history and examination still are going to be very important, because just analyzing the genomes also is not going to make sense till you have the clinical correlate. So they can, you have to have a very dedicated complete history and examination, that you can't just, you just can't have, you have to keep your eyes and ears open as soon as the child walks in or has brought into your outpatient, clinic, or whenever you're seeing, because everything, your history and examination starts concurrently. And I think that Dr. Mary would agree is going to continue to remain the mainstay, and one has to understand, and that, it should not be that you are just retrofixing after getting your, genetic analysis. You have to have a vision, have your thought processes, and flow chart in your mind, what you're thinking. And this is going to be supportive as of now. And because how many that is something very important, and we should not feel that you just have an MRI, you have a genetic testing, and forget about your history examination. This will not going to happen. History examination will remain important. Do you want to add something on this aspect before I ask Mahesh to make some comments, because I don't think anybody has put anything in the question.
One question, ma'am.
Yeah.
By Dr. Bandi Ramya. How to interpret presence of pathogenic variants in healthy individuals in these projects, like Indigen?
These are the carrier frequency. This is not the pathogenic variant. These are the carrier. So the pathogenic, these are the what I'm talking about, the healthy individual. This is in the heterozygous condition, not in the homozygous condition.
Mahesh, you want to make any comments?
No, ma'am. Nice talk, and it's nice to know that in India we're able to do so much, ma'am, actually, especially the functional studies. It's a good thing by IGIB that there's certain facility in India only. Earlier, it was also supposed to outside. So now in India also we can do the studies. Really nice to know.
Dr. Vinod, can you speak? Or your connectivity is...
Sir has logged out, ma'am.
Okay, it's logged out, fine. Any of the panelists would like to speak? Dr. Putin, Dr. Sanjay, Dr. Mary, and then we can have one take-home message from all of them. And then finally, Dr. Vinu is going to give the final.
If I can say something about what Dr. Putani just mentioned, and that the importance of genomics, and that's the one aspect that she mentioned about just helping solve a lot of cases. I work with my clinical colleagues from pediatric endocrinology, for example, and then we have been able to like identify the causal variants in many cases after doing whole genome sequencing of the patients and their parents or the siblings. And some of these cases actually, to the second point that Dr. Shefali mentioned about collecting the phenotypic information, the clinical history and everything, that's equally or more important actually in some of the times, because we have seen that sometimes with the genetic diagnosis, we are able to change the diagnosis that the clinician has made because they hadn't looked at certain specific aspects of the phenotype. So there was a specific case where we had to ask the clinician to go back and look at the patient again to see whether there is a specific condition in the eyes, and then we were able to they were able to confirm that. So it's now that we are going back and forth to find the potential causal variants and whether we can find the phenotype in the patient. So if we know all the phenotypic characteristics of the patient, then we can actually make a better diagnosis from the genome sequencing data. So thank you very much for the kind of vision that you have in India to move with the genomics and continue with taking the more accurate phenotype, collecting more correct phenotype from the patients.
Dr. Sanjay?
Yes, so my message is for clinicians. What I have seen that clinicians are very reluctant to do the genetic testing. And situation is a little different and better in apex centers like tertiary care center like AIMS, PGI Chandigarh or center we are working. But there is a sense of I will say complacency that they say इससे क्या होगा, इससे क्या फायदा होगा, अगर आ भी गया तो क्या फर्क पड़ जाएगा, देना तो वही ड्रग है. The question is that we need to understand that we are contributing to science, and unless until we don't make a inroads into the genetic diagnosis, the disease will be never understood. I will give you one example of a patient who was just saying that I have a left twisting of the hand, and I said that when it happens, he said that when I'm running. I thought, okay, he looks like a case of paroxysmal kinesiogenic dyskinesia. I got the genetic testing done. He was PRRT2 positive. So the question is that unless until you don't make an effort, the diagnosis will never be made. He will be continued treating as a case of epilepsy or maybe functional disorder. So the effort should come from the clinician, and we need to realize that this is important. And we need to collaborate with the molecular biologists, molecular genetic, and unless until we don't have our own data, how long we are going to survive in this world where the data is flooded from the Western countries? So I think we need to introspect, and as a scientific community, we need to collaborate more with the basic science people.
Dr. Mary?
I think both sides of the coin are important. As Dr. Sanjay would agree, when you have a functional movement disorder, there are people who send genetic testing. Functional movement disorders are a positive diagnosis, not a negative diagnosis. It is very easy to make a positive diagnosis of functional movement disorder and avoid spending money for costly tests. So also, if you have a spinal muscular atrophy, you should be able to distinguish that element disorder from a UMN disorder and avoid an MRI from vertex to coccyx. And that is very important. The clinical part is important. However, as he has rightly said, we should not go shy away from doing genetic tests whenever it is important, as when you know it is a genetic condition like inborn error of metabolism. And nowadays we are making diagnosis of 3-methyl-gluconic aciduria where we have a dietary treatment. All the other inborn errors of metabolism have some treatment now. And therefore, it's very, very important to send the genomics in cases where it is indicated.
Thank you. Dr. Binu?
Yeah, I would like to make three comments and one request. So the comment is that what India is lacking right now is, let's say, intake of treatable IND. So we don't have a pan-India study. So if we can actually make it groups like in pan-India and try to club each and every center representing each and every state in India. So that would be that data will be useful for like future next 2030. We are lacking that, but other countries are doing the same. So one thing is we can actually foresee that we could able to like gather different spectrum of the mutation from different state, and that data can be used for the like precision medicine. And second thing is whenever it's a harmony between clinician and the basic scientist. Whenever a clinician refer a case for a generic diagnosis, we would like to request them that detailed clinical history is needed, because for prioritizing the variant, that is more important. So when you actually detailed narrated, so let's say if a particular variant is the case is the detailed clinical history is available, so the prioritization of variant would be easy. Otherwise, actually, we have to do like unbiased test, then come into the analysis. So instead of avoiding that confusion, like from initial stage itself, if you have a detailed clinical history, then prioritization and diagnosis of the case would be much more easier than the other way. So that is my comment.
Mahesh, you want to say something? Mahesh? Dr. Mahesh?
No, ma'am. No, ma'am.
Okay, I think I want to thank everyone for having joined, and we end this series of what the experts say, and there were a lot of them were on genomics, and that is because that's the way forward for I think for any specialty now. But from next week we start the SIG, special interest group of the neuromuscular, and we'll be coming with a panel discussion next week on multidisciplinary management of SMA to begin with. Yeah, so looking forward for you all to join, and thank you so much. Thank you all. Bye.
Thank you.
Thank you all. Thank you.
Bye.
Good night.
Bye. Bye. Good night.
Good night all.